In Chapter 4 of our text, we're going to deal with the topics of enzymes, metabolism, and cellular respiration. These are very important topics and very interesting topics in the field of biology. And our text falls out pretty nicely and divides this chapter into two portions. The first portion deals with enzymes, the second portion deals with metabolism and cellular respiration. So that's how we're also going to approach these topics. We'll talk about enzymes first, and then we'll move on to metabolism and cellular respiration. Let's start with our discussion of enzymes. We first want to point out that enzymes are proteins. We talked about the 4 different types of biological molecules, and only one of those types of molecules actually acts as an enzyme. Acts as an enzyme. Enzymes are proteins, and this is going to be important as we continue our discussion of enzymes. Enzymes are proteins that speed up chemical reactions in a cell, and interestingly, every single reaction that occurs in a living cell depends on enzymes. We use the term metabolism to refer to all the chemical reactions that occur in the body, and each one of those chemical reactions depends on an enzyme. I want to skip slides at this point and come back to this one because I want to really start our discussion of enzymes by talking about what they do. So enzymes do, this is exactly what enzymes do. Enzymes convert substrates into products, as it says on our slide. And so this is how they do it. If we look at the diagram at the bottom of this slide, we see the enzyme depicted in green. Remember, the enzyme is a protein. And the enzyme has a particular spot on it that's referred to as an active site, shown at the top of this particular enzyme. There's also a substrate, and so the substrate is going to be the molecule that binds to that active site. And so you can see in this case, under number 1 there, the substrate binds to the active site, and the enzyme then performs its function. In this case, it's putting stress on that bond that's holding the 2 portions of glucose together. When stress is placed on that bond breaks, and then the products are released from the active site. The substrate is turned into a product. And so the products in this case are glucose and fructose. You dealt with these 2 molecules in your first lab, actually. So glucose and fructose are monosaccharides, and the original substrate, sucrose, is a disaccharide. And so this is how enzymes work. And there's a couple points we want to make regarding the action of enzymes here. One is that enzymes are very substrate specific. One enzyme will only catalyze one reaction. This means that one enzyme only binds to one particular substrate. And so when you think about the interaction between an enzyme and a substrate, I want you to not, this is an interesting thing to ask of you, but I want you to not think about the depiction that's shown on this slide here entirely, because just out of necessity, this depiction is dumbed down a little bit so that we can understand the basic idea of what's happening. In reality, the interaction between a substrate and an enzyme is very complex, and it is better modeled by a lock and key, what's described as a lock and key model. And so imagine that you took a key and you started walking down the main street in Wilmore and just randomly putting that key into locks in front doors. You wouldn't expect that lock. It's possible, but you would not expect that lock to open many, if any, front doors. And so that's exactly what we would expect when we just take a random substrate and try to put it into the active site of various enzymes. It's very unlikely that that substrate is going to fit into any of those active sites except for the one enzyme that it's supposed to bind to. And remember, that's all about shape. The shape of the active site, even though it's depicted as relatively simple in our screen here, is actually very complex. And so that's what makes that lock and key fit possible. So enzymes are very substrate specific. We also want to point out this. We understand the binding of enzymes to substrates by this model that we refer to as the induced fit model. The induced fit model says this, that when the substrate comes along and makes contact with the active site, it changes the shape of the active site somewhat and allows the fit to occur perfectly. And so interestingly, the shape, even though the shape is very specific, the shape is not perfect before that substrate comes along and binds to the active site. So the induced fit model says that the active site changes shape slightly to accommodate the appropriate substrate. I now want to take just a moment to show you one of my favorite depictions of the interaction between an enzyme and a substrate. And so on the screen we have the enzyme shown in blue, the substrate shown in green. You can see the active site of the enzyme shown as the indentation on the top of the blue enzyme molecule. And so what's going to happen is that the substrate is going to bind to the active site. the substrate is going to be transformed into a product, and the product is going to leave. Another reactant is going to come, it's going to be transformed into a product, into actually two products, and those products are going to leave. And so if we look at it again quickly, what we see is that the enzyme never changes. Multiple substrates come and bind to that active site and are turned into products, but the enzyme itself never changes. And interestingly, in addition, this whole process occurs very rapidly, even 1,000 times, up to 1,000 times per second, which is an amazing rate. But we have to remember that these things are very small and moving very rapidly. But enzymes can catalyze a large number of reactions in a very short period of time. The last topic that we need to deal with related to enzymes is how enzymes work. And the answer to that question is that enzymes work by lowering the activation energy that's needed for a chemical reaction to occur. This is depicted pretty nicely in the diagram provided by your text that shows up on the bottom portion of this slide. In part A of this diagram, we see that the reactants, which in the case of the enzymatically catalyzed reactions, as you already know, are substrates. So remember, reactants and substrates are equivalent terms in the case of a reaction that's catalyzed by an enzyme. So the reactants and the products in this reaction are depicted by people. And so we can see in the diagram, the part A portion of the diagram, that the people that are reactants want to become products. And they have a certain amount of energy, but they don't all become products because of that barrier that stands between the reactant and the product category that's referred to as an activation energy, or an activation energy barrier. So the people that are reactants, they jump around, they have a certain amount of energy, but very few of them can jump over that barrier and become products. They want to do that, that's the natural tendency, but they can't because of that barrier. What an enzyme does is this. If we look over to part B, we see that that barrier is actually reduced. In other words, the height of the barrier is lowered, which I encourage you to look closely to see. I realize that I'm not sure exactly why, but it's still drawn about the same height. But you see that that little red portion represents a bar that goes across, and so it's like a pole vault sort of situation. There's a bar that's lying across there, and we're looking at it from the side, and so indeed that barrier has been lowered. And this means that now most of the people that are reactants can jump over that barrier and become products. And so this is a pretty good analogy to what actually happens in an enzymatically catalyzed reaction. The reactants will become products or they would become products, it would be more accurate to say, if that activation energy barrier was not present. What an enzyme does is it lowers that barrier that prevents the reactants from becoming products. And so a good question would be, how does this occur? And the answer to that is as follows. If we go back to this slide, we can see that what the enzyme does is that it provides a little microenvironment in which the reaction can occur. It puts stress on that bond, for instance, in this case, and it causes that bond to break. That bond would rarely break if the enzyme wasn't present, but when that substrate fits into the active site, then the enzyme twists the bond in just the right way that it allows that bond to break.